We work in the immune mechanisms of lung injury, repair and fibrosis. We want to try and understand which immune cells and how they contribute to progressive fibrosis. So how these immune cells make fibrosis worse relentlessly. And ultimately what we want to do is try and target these immune cells therapeutically and to design better clinical trials so that we bring better treatment to patients with fibrosis. In terms of techniques, overall, we use the best methods and best technology to answer our questions. We are working intensely on spatial analysis of cells within the lung tissue and single-cell transcriptomic analysis of lung cells. So our work spans from the molecular pathways in immune cells to murine models to highly translational studies, for example, trying to understand how we can measure inflammation in the alveolar, and we can do this by developing methods which are non-invasive. In the last few years, we've really enjoyed working with the WIM computational team on spatial analysis of the lung tissue using imaging mass cytometry. We use a combination of 37 metal-tagged antibody where we are able to give an identity to every single cell in severely diseased lung tissue. These methods have helped us understand what's truly happening in the lungs. So, for example, we've always known that patients with severe COVID have a phenomenon called emergency myelopoiesis in the blood, where we can see immature neutrophils and immature monocytes in the blood. But we never really knew what was happening in the lungs. And with our collaborators in Spain, where we got some very fresh lung tissue, and using this method that I've just described, we were able to see that these immature neutrophils actually go into the lungs. But only here, our tools were able to show us that they coalesce with other immune cells to form foci of inflammation around the stem cells of the alveolar unit, so probably impinging on the ability of the lungs to repair itself. In the area that I'm interested in, that's mechanistic science applied to human diseases, the most accurate picture has to be built by a team that comprises people with many different expertise and understanding of the problem. So now we have access to a wealth of data from single-cell work, but coming to a message, a true signal, requires all these people to work together as a team. And the challenge is really to get everyone to see the big picture, but also to drill down into the hard work and the good quality studies and good quality data that has to come through. So this takes time, commitment, and teamwork. And when it works, it's the most rewarding of challenges. Begin to see patterns emerging and discover a connection between different molecular pathways, for example. Currently, we have a great team of very bright, young and older scientists. We strive for a high standard of work that can be repeated, not just by us, but also by other scientists. We do take a little bit longer to work at this and to get to that point, either by making sure that our data has been turned over a number of times, or that our patient population is large enough to derive the right conclusions.